from your Human Biology book, Chapter 1. And I'm going to look at the goals and a few of these key terms for Chapter 1. So initially, we do want you to understand characteristics shared by all living organisms. So this is right from your textbook. So you can see that as I list these, you have descriptions of things that without characteristics that without being able to do these things, use materials and energy, reproduce and make more of a species, grow and develop, respond to their environment, and be organized, in terms of atom all the way up to the biosphere, that living things would not exist. So this word homeostasis here, as a quick recap, homeostasis happens in all organisms. In humans, it is the ability to maintain a constant internal environment. So we'll be making changes based on the needs of the body. More water, less water. Increased temperature, decreased temperature. More hormones, less hormones, etc. So that is homeostasis, maintained mostly with your nervous system and your endocrine system. As we discuss the levels of organization that we see in living things on Earth, this is, of course, the first level here. So living things are organized first by atoms. Non-living things are also made of atoms. But as we develop into living organisms, those atoms must become molecules that then collectively come together as a unit called a cell. You have multiple cell types, they become tissues, and so on. Then as we increase the levels of organization, those groups of tissues become organs. And so you're seeing a combination of nervous tissue creating the organ known as the brain. So it's not just a neuron like you see here, but you have multiple types of nervous system cells that make the brain. So then that's the organ, multiple tissue types making an organ. Then you have the organ completing, for instance, the organ system of the nervous system, and then the complete organism with all of those working organ systems. So human biology stops there, but we have the interaction with our environment. So first we look at a collection of the same type of organism, and that is considered the population, same species. So students will get mixed up here. Same species, that's the population of humans, the population of squirrels, the population of trees, and so on. Now, when you have a community, you have these populations interacting. So you have the species of humans, you've got trees. So that's a community. And then as we increase complexity, move that out of the way, you have the combination of the community of living things with the physical environment that those living things are in. That's the ecosystem, excuse me. And then the very top level is the biosphere. So be able to put these in order and understand their definitions. All righty. Next, as we look at the process of science, what do we know about science, or the science as a process? So as I list here, we know that when we say science is going on, it is really a process of knowing about the natural world, what we see, what we experience, what we perceive. So it is a process of learning about that natural world. So that's science. So understand that in order for it to be science, scientists and the process they use has to be objective. It has to end up with conclusions that may be modified based on the data that comes in. So as we compare the process of science, in which case we use the scientific method, we do want you to briefly understand that the process involves, let me move on to these steps: an observation, So you are observing the natural world. Again, the process of science, scientific method. Number two, based on your observations, you propose a hypothesis. Based on this, I believe, and I create a testable statement. That's important. And that testable statement, then known as the hypothesis, is put to the test. What's the test? That's the experiment and or observation or combination thereof, in which case, of course, you will collect data. Those data points or that collection of information is gathered up into results. The results of your experiment will include the data. All right. And then you take those results and data and you analyze those. That becomes your conclusion. Results analyzed, hypothesis is either supported or rejected, or maybe there's not enough information and you may have to circle around and revise that hypothesis. If a scientific theory is established, that means there are going to be many observations, many experiments, many data points, large numbers that help support the hypothesis. As you consider the process of that experiment and gathering information, a couple of key terms, understand that in a controlled study, there are going to be particular experimental variables and control variables that help you give you something to which to compare. So an experimental variable is the one that is purposely changed or manipulated. That's the experimental variable. The other variables around that experimental variable need to stay constant. So helpful to have the same gender for a particular study, to have the same pH, to have the same temperature. Then the groups are put into experimental groups and control groups. So understand that the control group is absolutely necessary for comparison to the experimental group. The experimental group has been exposed. Think experimental group exposed to the variable. The one that's the control group was not affected by that. So read about that in your text. And there's a good example of bacteria in the gut, and it may help you understand the difference between those two. As you consider reliable scientific information, remember that scientific journals are the ones in which the information has been put through the scientific method requirements, large studies that give you information that has been tested or vetted, but often hard to understand. Secondary sources then will, whether it's Time Magazine or a website that you look at, they will read those scientific articles and then give the interpretation of that. But you should be wary of the information that might be taken out of context if you're not looking at the original source in the scientific journal or reporting. Most of you are, I'm sure, familiar with graphs, but understand that when we're looking at a graph and we see that y-axis here and the x-axis here, be able to interpret that information and know that here, as an example, blood cholesterol levels going from 150 to 225, as that relates to a sequence, a chronological period in which we're seeing a variation in blood cholesterol levels in a particular study. So these are the two locations. You want to understand the data points that are represented there. As I stated in the beginning, science is a way of acquiring knowledge about the natural world. So that is a process that is separated from ethics, religion, aesthetics. It is hard to separate those for any one particular individual, but to define science is to say there's a process of observing, evaluating, reporting what you see. Technology is when you apply the scientific knowledge that you've gained through the process of science. Applying scientific knowledge for human interests. So understand the difference between those two. Next, we'll look at some of the topics in chapter 2, some that students might be mixed up a little bit on. Chapter 2 is, of course, chemistry, and it can be challenging if you have not studied chemistry in an earlier class or recently. So just do the best you can. As we move on to define what makes up the world, and then, of course, what makes up us, we start with this word matter. And anything that takes up space and has weight is matter. So organic things like humans, food, trees, and inorganic things like the mouse right here, right? They're all matter, including the air. When we consider elements versus atoms, so an element cannot be broken down by simple chemical means. And so you've all seen the chart, periodic table of the elements: carbon, oxygen, nitrogen, sulfur, gold, plutonium, uranium. These are all elements we find naturally. The atom is going to be the smallest unit of the element. So we find the element in nature. The very, very smallest particle of an element is an atom. We bind atoms together, then you have molecules. At least 2 or more atoms connected makes a molecule. All right, so one atom plus one atom makes a molecule, or hundreds or thousands of atoms make molecules. Funny joke for you, do not trust atoms. They make up everything. Here is a brief example of the periodic table showing some of the elements as they exist on the table. You do not need to be able to read this table per se, but it gives scientists information about each atom. What we find in atoms are some basic particles. We call them subatomic particles. So on your outline, just describe the structure of an atom. So let's look at carbon down here as an example. So atoms will have protons, neutrons, and electrons. A neutron is neutral, so we won't represent it with a charge at all. Neutral, 0 charge. Protons have a positive charge. All right, each proton gives the atom 1 positive charge. And those protons and neutrons are located in the nucleus, right? So here's a nucleus here. And for carbon, there are 6 protons and 6 neutrons. There is an additional particle, so 3 total, and that's an electron. Electrons are represented out here. In this case, electron shells. Electron shells. You are not going to have to determine how one particular atom is going to combine with another. But that is the basic organization of an atom. As you consider the term isotope, an isotope is a unique atom. In particular, isotopes can be used to use in medical imaging, which is what you're seeing in these pictures here. So by definition, be able to match up the fact that an isotope will always have the same atomic number. Atomic number is the number of protons. So let's say it's carbon. And carbon always has, here we go, protons. Always has 6 protons, but it will have a different number of neutrons. Remember how protons and neutrons are in the center of the atom? Let's go back to this picture. Protons and neutrons in the center. Well, if instead of having 6 neutrons, we end up having 8 neutrons in the middle, that's a heavier element. And it's going to create an isotope of carbon. It's called carbon-14 as an example. Carbon-14 is what we use for radioactive dating of certain types of organic material. Radioactive isotopes like carbon-14 are going to be, for instance, dating objects. But the main thing to understand is that you can use radioisotopes that are safe radioisotopes for medical imaging like X-rays and MRI, PET scan, and sometimes they can be used to kill cancer cells. Radiation that comes from a radioisotope can be damaging if it's a long period of time that you are exposed to the radioisotope, or if it is something like a very, very powerful radioisotope like uranium or plutonium. All right, I've forgotten the number. I think uranium-3. I'm not going to tell you, actually. I don't remember. But those are the kinds of radioisotopes that are very, very harmful. As we talk about what creates molecules, we know at least 2 or more atoms combined. So they're bound together, chemically connected. They can be made of the same atom, like oxygen that we breathe is made of 2 oxygen. We call it O2. Or water is made of different atoms. 2 hydrogens, H2O, means 2 hydrogens and 1 oxygen. When we talk about how these chemicals are combined, one way of combining chemicals to make a molecule is an ionic bond. Ionic bonds will have the situation where there's actually a donation or a taking away of electrons. That's all you really need to remember, to gain or lose. So what happens in this example, sodium is going to give away its electron to chlorine. When that happens, it will create an ionic bond. There's an actual loss by sodium, a gain by chlorine, and now it becomes salt, sodium chloride. And here's a salt crystal. And that would be ionically bonded. These 2 molecules here, sodium and chloride, or excuse me, they're actually called ions. I'm going to list that here. So when the electron is given up, sodium becomes positive because it lost one of its electrons. Chloride becomes negative because it gained an electron. Electrons have a negative charge. And so that will happen. These ions are created if we had a beaker of water and we took one of these salt crystals and dumped it into the water. All right, so here's our beaker of water, put salt in the water. So what is sodium chloride, the molecule connected by ionic bonds, splits up and becomes a sodium ion, Na+, and a chloride ion, Cl-. So those are called ions. It's a particle that has a charge on it, a plus charge, a minus charge. Those are called ions. There's some other examples, but we're going to go on to covalent bonds. So listed here, a covalent bond is not a complete giving up or gain of electrons. Covalent bonds, you see shared electrons. So there's your key, shared electrons, and you can see an example of water. Here's the oxygen is here, the hydrogens are out here, and they are going to share the outermost electron. So that's a sharing. It's not a giving up of the electrons. You'll want to review the properties of water very briefly. So listed here and also in your lecture textbook, we know water stays liquid at room temperature. It does not change temperature quickly. It will require high heat to vaporize, to go from a liquid to a gas, like when you boil water. Frozen water is less dense than liquid water. How do we know that? Got a glass of ice water, All right, here's my water. I'm going to put in an ice cube. Ice cubes do what? Ice cubes float. Think of an iceberg. Iceberg rests on top of the liquid water. So frozen water is less dense than liquid water. We know that water molecules will cling together. We'll look at a picture of that in just a moment. And it is a good solvent for dissolving things. So we're made of 66% or close to 70% water. Humans are. So the bond that will help connect water molecules is called the hydrogen bond. So this is our third bond type. We talked about ionic bonds, covalent bonds. Hydrogen bonds mean we have a hydrogen here, put an H, and it is attracted to the oxygen, which is here. This is an oxygen. All right, so as water molecules sit next to each other in a liquid state, you're having this relationship of the oxygen turning towards a hydrogen. So you're going to see what is called a partial negative. I think I have another, no, I don't have another picture. And so the oxygen is partially negative and the hydrogens are partially positive. Without getting into the theory of why that happens, know that because of this lopsided electricity, you're going to have water molecules have hydrogen bonding. So every one of these dots, dotted lines, is representing the attraction of a negatively charged oxygen attracted to a positively charged hydrogen of a different molecule. They are relatively weak bonds compared to ionic and covalent bonds. Next, we look at acids and bases. And not a lot of detail you need to understand about this, but as we look at the pH scale, let's see, here we go. When we're looking at a pH scale, what is being measured on the scale, because scales measure things, is the amount of hydrogen ion. That's a hydrogen atom that has a plus charge on it. That's a hydrogen ion. So that's what we're measuring. So as I look back at the definition for acids and bases, acids are chemicals, compounds that will release hydrogen ions. So they tend to put that into the solution. So it's a chemical like muriatic acid that releases hydrogen ions. And so that it ends up putting these into the solution, and pH will drop. It lowers the pH. Acids tend to have a sour taste. As opposed to bases, they're not going to release, they're going to take up hydrogen ions. All right, and so when you put those into a solution, they're going to increase the pH. So that's the bottom line. And you'll learn later on in this semester that the pH of your body, your blood, your fluids in your brain, fluids in your lymphatic system, the pH is very closely monitored. And finally, instead of being sour like an acid, think of lemon juice, very sour, it's acidic. Bases are bitter and tend to be a little more slippery. Here is an example of some compounds on the pH scale. So pH value of 2 as an example, very acidic. Stomach acid, lemon juice, very acidic, as opposed to household ammonia, very basic, a strong chemical that has a high pH of 11. All right, and you can see that without getting into how they came up with this scale, understand water has a pH value of 7. All right, so when we talk about buffering the pH, the buffer will keep pH in normal limits. All right, so the buffer can help it from making drastic changes, and we have buffers in our body to do that. So a picture of Rolaids. So if you had an acidic stomach, you're going to take a buffer like Rolaids. We have done tests in biology class and we've learned Alka-Seltzer actually works much better than Rolaids when we do it in the biology lab. But either way, it would help to increase the pH if you have a very acidic stomach acid. When we consider building or breaking down large molecules, in particular, organic molecules are going to be large. Organic molecules will always contain the element carbon, as opposed to inorganic molecules like salts, acids, bases, water. There is no carbon, or very rarely carbon, but organic molecules are carbon-based. If we were to build up, make a larger organic molecule, we'd take two separate units. Here's one, and here's another. And we're going to do what's called a dehydration reaction. And now we've built a molecule that has two subunits attached. When that happened, you see a product that ended up coming out, water. Or dehydrating in order to build. Sometimes this is called a dehydration synthesis reaction. So your textbook is calling it just a dehydration reaction, but dehydration synthesis can help you remind you that it's actually building bigger molecules. If we were to take apart this molecule, we're going to do the opposite. So here's the larger molecule down here. We're going to use the reaction called a hydrolysis reaction. And with that, we add a water molecule. It's not a product. We're going to add the water, take pieces of the water, and use it to split the larger molecule. So now we have this separate unit from this one. That's hydrolysis. Same process is used to build or tear up carbohydrates, proteins, lipids, nucleic acids. And that would be our list of 4 main organic molecules. Your 4 main classes of organic molecules: carbohydrate, lipid, protein, and nucleic acid. As we consider the first group of organic compounds, known as carbohydrates, we'll see that they build into larger molecules, starting with a molecule called a monosaccharide. Mono means one, saccharide is sugar. And of course, we know sugars are part of the category of molecules known as carbohydrates. So these single 1 units called monosaccharides are built into larger molecules that we're going to call disaccharides. All right, what are carbohydrates important for? Short and long-term energy. The sugars that you have in a Snickers bar, quick energy in an orange juice, versus long-term energy when we eat starches and store starches. Here would be the simple carbohydrate called a monosaccharide. So you've got a single one here. And then when we put the two together, when we take 2 monosaccharides, we have now a disaccharide. So that's the main thing to understand. Mono 1, di 2. So as we build carbohydrates, if you have a complex carbohydrate, you can see clearly chains of a single monomer over and over and over again. In this case, this would be chains of glucose, a sugar molecule. So polysaccharides are a type of, are also called complex carbohydrates, many carbon rings over and over again. You should understand that glycogen is one of these complex carbohydrates that we store in our body. Mainly we'll store it in muscle and liver. So that is a polysaccharide that we store, energy that is stored. Now this is not fat, this is a carbohydrate. Starch is also a polysaccharide or complex carbohydrate but that is stored in plants. So here you're seeing starch grains. Here's a grain of starch that's in a potato as an example. All right, so moving on to our second class of organic compounds, carbohydrates, now lipids. Lipids we know don't mix with water. They don't dissolve in water. Think of fats and oils that don't mix with water. Oil and vinegar dressing doesn't mix. We do find them to help with energy. That's why we store fat in our belly and all over the body, and found in cell membranes as an example. So the main categories, fats and oils are kind of in one group, phospholipids, and then steroids. These are types of lipids. Let's take a look at the category of lipid known as fats and oils. Main difference here is that while they have a similar structure, fats are animal origin. So for instance, bacon fat. You cook bacon, the fat in the pan cools and it becomes solid at room temperature, right? So then we also have animals, of course, store fat for long-term energy storage. We can access that, but also insulate, cushion organs, have other roles as well. If it's considered an oil instead of a fat, similar structure, but oils are plant origin. And when you cool down an oil, it doesn't become solid. Remember, it is liquid at room temperature. So a quick visual of how a fat or oil molecule is built. You'll have a glycerol. You won't have to recognize these shapes, but you put a glycerol with 3 fatty acids. Now you have a fat or oil molecule. So this would be known as a triglyceride, tri for 3 fatty acids. And you may have heard of triglycerides being measured in when people have a blood test as an example. The next category of lipids, still under lipids, after fats and oils, we have steroids. Steroids are a cholesterol-based molecule. So here's a cholesterol. It has these 4 rings. And then we use that cholesterol to build things like testosterone or estrogen. So examples of the sex hormones. You like my choice for pictures here. So on your definition for steroids, remember it's still a lipid, but it's not a fat or oil. It has a different structure. These carbon rings that are fused, 4 carbon rings, and cholesterol, which we find embedded in membranes and other kind of structural roles. And then we have multiple types of hormones that have the same shape. An example would be the reproductive hormones. Moving on to proteins. On your study guide, we look at proteins being our 3rd category of organic compounds. It's important to understand the unit is not a sugar unit. It's not made of fats or fatty acids. When you build proteins, you're building amino acids in a chain. All right, subunit, or sometimes we call subunits monomers. If you see that monomer and subunit, it's really referring to the same thing. And you have these repeating monomers or subunits of amino acids. What are proteins important for? Quite a few things. All right, proteins kind of run the show. Hormones, enzymes, antibodies, transport molecules, in addition to building the body, skin, bones, muscle, membranes, many places we have proteins. Proteins can change shape in a process that we call denaturing or denaturation or naturation. So when it changes shape, it can lose the function. So an example would be an enzyme that has now changed shape. It doesn't fit the molecule it's supposed to help activate, and it doesn't work. Antibodies, we know, are important for immune function. If that changes shape, it can't identify the microbe, the thing that we need to take care of to get rid of. So those are some roles for proteins. When we build proteins, we've stated that they're made of amino acids, and there are only 20 different amino acids that exist. Here are a few examples. You don't have to know structures, but each one is similar in 3 parts. These 3 parts right here repeat. So you have a carbon in the middle, an amino group here, always at 1 hydrogen, and a group that we call a carboxyl group. And then the light blue here, you can see that these parts are how each amino acid is different. All right, and again, you have 20 different R groups. This is an R group. So we have 20 different amino acids. We put them in chains to build these large molecules called proteins. So on your checklist, your study guide, you see that as we build proteins, there are actually 4 different levels of putting a protein together. You start with the primary level, which means you're going to put the amino acids in a particular order. The order really comes from the fact that you have a DNA molecule that will have the code. It's going to say, this is how you should put those amino acids together. So we're not going to go through that. Primary chain of amino acids, that's how we start. What order are they in? Secondary. Now that chain of amino acids begins to fold. And so you're going to see things called, I think it's called an alpha helix. So I'll put the word here. And then there are shapes known as beta pleated sheets. Beta. So these 2 shape descriptions are the secondary level. You see twisting, you see up and down structures. Third level called tertiary. Tertiary level means now it begins to have a 3-dimensional shape, really as it interacts with water. So that's the singular protein by itself. Occasionally, there's a 4th level called quaternary, and you would combine 2 different tertiary structures or 4 different tertiary structures to make the protein. An example would be this building up of, here it looks like a hemoglobin. So here's primary structure, chain of amino acids. Secondary structure, begin to see the spiral or these funny foldings called beta sheets, beta pleats, pleated sheets, I guess. Tertiary, that 3rd level, is when we begin to see a 3-dimensional shape, even more so. And then sometimes you get to a 4th level where here you can see 4 different tertiary structures that come together. And this would be what hemoglobin would look like. Here are some examples of quaternary structures where you have a muscle protein, or you might see, let's see, keratin that you have in the skin and in hair. And here's how hemoglobin is built. You've got a tertiary structure, a single unit that comes together with 3 other subunits to make hemoglobin, which carries your oxygen in your red blood cells. The 4th category of organic compounds is a group known as nucleic acids. Nucleic acids, you have seen this twisted ladder shape. And of course, you hopefully by now you know that it's DNA. So when we build nucleic acids, DNA and RNA, remember DNA standing for deoxyribonucleic acid. That's this one. RNA is known as ribonucleic acid. Those are both nucleic acids. To build them, you'll have a subunit called a nucleotide. Not a lot of detail you need to understand about those, but DNA and RNA have a role in directing and producing proteins. DNA is the original director, all right, office. So this is the president in terms of directing what proteins are going to be made. RNA is really part of the machinery to make proteins. This is a nucleotide. So the main thing to understand is these units repeat over and over again, millions of times. DNA is the largest, by far, the largest molecule in the body. So nucleotides will have a phosphate group, a sugar, the S is sugar, and a base. Oh, I forgot that label was there. So that repeats over and over again. When you see the repeating unit, you're going to see here, here's a phosphate over here. Here's a sugar. And here's the base. In this case, it's the base that starts with a G called guanine. The main thing to understand is you will see whether DNA or RNA, there are always 4 bases. There exist 5 different types of bases. The RNA is the only one that has uracil. That's what you see in RNA. Like here, you're seeing uracil. And DNA has thymine. But each molecule, you're going to have matching 4 different kinds of bases. You do not need to understand how they come together, how they pull apart at this point. All right, and then a summary here of how those 2 molecules are different. Remember, DNA is huge, right? Very, very big. It's got a different sugar called deoxyribose. The bases are A, T, G, C, and G, whereas RNA, the sugar is ribose, and you can see it has a U instead of the T. So the base switches out. This is the one that's double-stranded. So you'll see the thing that looks like the ladder, right? Like that, that's DNA. RNA is single-stranded. All right. And now finishing up the review with chapter 3, cell structure and function. And there'll be quite a lot in this chapter that is not on our review because we're not getting into a lot of the details. But to begin with, cells come with the choice, so to speak, to be either a prokaryotic cell or a eukaryotic cell, prokaryotic and eukaryotic. So prokaryotic, think of pro before or first, thought to be the first cells that existed. The key here is that prokaryotic cells do not have a nucleus. So they'll have a barrier, they'll have a membrane, but the material that's inside is randomly organized. All right, so there is not going to be a specific nucleus. Eukaryotic cells, on the other hand, do have a nucleus. So when you look at a eukaryotic cell, which is of course what we have, the nucleus houses the DNA, and it is much more organized as a cellular machine, so to speak. Many different membrane-bound organelles of which you've already studied, and they're going to be located in here, sometimes a series of membranes, sometimes specific structures like little mitochondria that have inner membranes, sometimes little vesicles. Those are organelles. And so eukaryotic cells will look like that. Here are some actual prokaryotic versus eukaryotic cells. So prokaryotic cells are types of bacteria, as an example. And they, viruses would not be considered bacteria. Viruses are actually not living structures, but an encasement of nucleic acid that invades cells, mostly eukaryotic cell organisms. So prokaryotic cells, here would be a simple rod-shaped bacteria or a spiral-shaped bacterium. They do not have nuclei, and they're much more simple in their organization. Eukaryotic cells, you can see this complex web of these membranes in this one. Very large nucleus. And here is also a eukaryotic cell shown with a scanning electron micrograph, having much more complex organization. So in common, we know that prokaryotic and eukaryotic cells have a barrier, right? They've got to have a boundary, plasma membrane, and they've got interior material that we're going to call cytoplasm, which tends to be kind of jelly-like. They're going to call that semi-fluid type of texture. And of course, both prokaryotic and eukaryotic cells need directions. So they have DNA molecules in them. I'm going to focus next on eukaryotic cells, since this is a human biology class, and the focus will be on these more complicated cells. You will not be given a photo or diagram and need to tell me the name of any particular structure. All right. Your test is verbal, written, it is multiple choice, and occasionally true false. So you're not going to be seeing figures to label. I'll begin looking at structures of the cell by focusing on the cell membrane or plasma membrane. So we're seeing just a little piece of it here. And characteristics of the plasma membrane for you to remember. The general makeup is what we call a phospholipid bilayer. So these little molecules here that look like hairpins, they draw them like this with two tails. And that's known as a phospholipid. And you've got one layer there and one layer there. So entire membrane has this very fluid, movable phospholipid bilayer. You can see embedded in that bilayer are many, many different proteins. So these can be channels, they can be anchors, like this one creating an anchor for this other protein there. And then you also see oftentimes cholesterol that exists, and I don't believe it's barely labeled here. Cholesterol would actually kind of stiffen the membrane. And then occasionally you will see carbohydrates that are attached to the proteins. Here's a carbohydrate chain. It's attached here to the protein, and it actually makes the cell kind of sticky on the surface. In addition, there can be some additional lipids. So remember our categories of organic compounds: proteins, carbohydrates, lipids, and nucleic acids, which would be DNA and RNA. You don't see DNA and RNA in the plasma membrane. Here is an enlarged view of that membrane. And what you should understand is that the membrane's role is it is a boundary, it's a barrier, but the cell can pick and choose what comes across, right? So here is a protein that's going to act as a channel for some molecules, maybe molecules that can't normally cross the phospholipid bilayer because they are not lipids. They're not lipid-like, they can't come across. So for instance, if it was a glucose molecule out here, a G for glucose, glucose cannot get through this membrane. All right, so it's going to require a transporter to move it in. So membranes are what we call selectively permeable. So just understand the description means we're going to select what can permeate. And this is a really helpful figure that you've seen in your book and in your PowerPoint and in your Connect homework assignments, showing you some charged molecules, ions, large molecules that can't get through, water requiring a special channel for it to travel through. At the same time, there are other molecules that we'll call non-charged molecules, like lipids, things that are very lipid-like, can get right through, back and forth, the phospholipid bilayer. All right, so I'm going to very briefly go over the processes that help move things across the membrane: diffusion, osmosis, facilitated transport, also called facilitated diffusion, active transport, and endo and exocytosis. These two here are going to be part of processes that require energy. So they are active membrane processes. All right, so that's 4 and 5: active transport, endocytosis, exocytosis in the different forms. They require energy. So active processes require energy, and we're going to say that it's requiring energy in the form of ATP. All right, active, need the high energy molecule called ATP. If it is one of these three, just as a quick summary, those are passive processes, and that means that they are not going to require ATP, whether it's their structure or the use of a concentration gradient where things get pushed across. No ATP is required in a passive process. All right, so again, what are these? They are different ways to move things across the cell membrane or the plasma membrane. With the two processes known as diffusion and osmosis, what you want to understand, remember I just said these are passive processes. They don't require the addition of an energy molecule called ATP. And with diffusion, you have random movement going across the membrane, and it's always going to be from high to low concentration. So you've got crowded molecules on one side of the membrane. Remember, here's the plasma membrane, and you're going to have those molecules move across. So here, well, we'll just move on from there. If it is osmosis, we mean specifically the movement of a water molecule. So that's representing a water molecule, and it's moving across the membrane because there's a lot of water over here and not so much over here. It's going down its concentration gradient. It goes from where there's a lot of water to not very much water. In terms of the molecule that is a random movement of molecules across the membrane, in this case, we're looking at these purple molecules. All right, that's not water. It's what we call the solute, the dissolved particle. And it's going to move from more so, and you can't really tell if this arrow is bigger, but that's what it's representing, that it's going to move more into the cell because by diffusion, there are more molecules over here, a higher concentration of these purple molecules. And it just will diffuse, if it's able to, diffuse across the membrane more into the cell than it is going out of the cell. So that's the difference between those two. When we see osmosis being important in the human body, we're going to see a term called tonicity representing whether or not the solution is appropriate for the living cell. So in this case, we're going to say that a hypertonic solution that a cell sits in, and the example is over here on the far right, a hypertonic solution. What will happen if cells are sitting, our cells, as an example, these red blood cells are sitting in a hypertonic solution? The cells are going to shrivel up, and you won't be having to write an essay or describe why this is happening, but you can see if water moves out, it's leaving the cell by osmosis because of this concentration of particles out here. A high concentration of salt. Let's say you put these cells in salt water out of the Gulf. These cells are going to shrink. That's hypertonic. If we put it in these cells in a hypotonic solution, the number of dissolved particles here is not as much as what's in the red blood cell. So water will tend to move in. That's hypotonic solution. And they become more round. Oh, that's how I always remembered it, hypotonic. And they puff out and usually will actually burst open because water moves in so much. And then finally, an isotonic solution is going to be, iso means same. So the concentration of dissolved particles out here that are dissolved in water is the same concentration as what you have in the cell. And this is, of course, what we want our cells to be bathed in. So the cell doesn't shrink, the cell doesn't blow up. That's isotonic solution. To compare another transport or way to get materials across, you recognize cell membrane here with phospholipid bilayer. You have a protein, this orange molecule is a protein that's going to help move molecules across. Facilitated diffusion, you remember diffusion means we don't need any ATP. And in that case, the transport of molecules goes across the membrane. It always goes from high to low concentration of that particle. It's diffusing. It's going down the gradient. I like to say it's kind of going downhill. So any diffusion is always going to be down the gradient from high concentration to low concentration and will not require ATP. All right, so facilitated diffusion does not require ATP. It does require a protein carrier because whatever the molecule is, in this example, we're talking about, for instance, well, I'm not going to talk about that. Let's move on. Next one, to compare that to active transport. Remember, I said you have to use ATP to pump something across the membrane. So that's what's demonstrated here. ATP is an energy molecule. It actually transfers one of its phosphates. You don't need to understand how that happens. But because of that, it's going to help push out molecules. In this case, it's going to push out 3 sodium molecules, pump it out, even though there's already a bunch of sodium out here. It's going to pump it against the concentration gradient and push it out. Key is active, uses ATP. Does this need a protein carrier? Yes. Also needs a protein carrier, but it's using some energy in the form of ATP to move the molecule. So this was an animation that I won't play for you, but understand this is a similar setup where you've got a protein carrier, molecules that can be moved. So finally, on the movement of substances through endocytosis and exocytosis, these were part of that active process. These are big movements, bulk movement, we call it. So if it's endocytosis, that root endo tells you we're taking within. So endocytosis, we take molecules from the outside, move them in. We actually create a small ring of the membrane around the molecules, and then it comes in as this thing called a vesicle. Endocytosis. Exocytosis, exo is the root that tells you exit. Exocytosis, notice the arrows, the direction. We have a package of a product right here. We're going to move that product to the edge of the membrane, and then these molecules that are here, what do? They're being released in that space. All right, that's an exocytosis. These will require multiple molecules of ATP. There are pulley systems and all kinds of more complicated mechanisms to get these vesicles in and out. All right, so we're going to talk very briefly about structures involved in making proteins, but I'll make this pretty brief. So we'll begin with where the instructions originate to know how to make proteins, and that's going to be in the nucleus. So cells have one nucleus for the most part, and it's this entire thing is the nucleus. And it is porous. It has these little openings that allow molecules in and out. We know DNA is inside. DNA, remember, contains the genes, the directions for how to make proteins. When you put DNA together with proteins, you call it chromatin. Chromatin. So that's what you're seeing. In fact, they have not labeled very well. Chromatin would be a lot of this coiled material, like filling up the nucleus. That's chromatin. And then the space in there is called nucleoplasm. So the important thing to understand is that the nucleus works together with these structures called ribosomes. So in this picture, the ribosomes are represented by all these little yellow dots. All right, so ribosomes are on the outside of the nucleus. Ribosomes are attached here to an organelle called endoplasmic reticulum. Those are ribosomes. So not important that you know what they're made out of, but we know they locate in two places. They're either attached, fixed to the ER. So that's what you're seeing over here, or they're floating around in the cell. So you'd actually see ribosomes floating all over here. They're showing them on the surface of the nucleus, but all around. So what's their job? Making proteins. So they're part of the machinery to make proteins. The relationship of this thing called an endomembrane system and the proteins is that this is really the inside the factory. These are going to be the conveyor belts and the collection sites for the proteins that are made. All right, so it's going to be a combination of these structures that make an within endo in the cell membrane system. All right, and really the main thing is to understand the visual of what happens when a protein is made. We got some directions that came from the nucleus, from the DNA. We know that those directions go out to where the ribosomes are located. They're able to crank out protein production. So for instance here, there we go. Rough ER, which means the structure that is the membrane freeway with all these little yellow ribosomes, synthesizes the protein. Now it's going to ship it into these vesicles. Those vesicles join up with another structure called the Golgi apparatus. Golgi finishes up adding, subtracting, cutting up parts, and then that will create another vesicle that then gets kicked out. So that process, this is all exocytosis, involves the endomembrane system. Here is an incoming structure which doesn't relate to making proteins, but just to identify that in fact this is an endocytosis. And we'll just leave it at that. All right. So what makes up the endomembrane system? You saw it on the earlier picture. These are the main structures. Rough ER or endoplasmic reticulum has ribosomes. That's why it is rough. Smooth ER does not have ribosomes. It's going to have a different job working with the metabolism of carbohydrates and lipids. Golgi, you saw in the picture, flattened membranes that help in processing and packaging up the product. And then of course we deliver those. And the lysosome, if you see a description of that on the test, you know that these are little vesicles that can help digest what has come in. So they're all part of this, what we call the endomembrane system. The structure in the cell known as the cytoskeleton, of course, is given that name because it's going to give the cell structure, but really has nothing to do with bone. This is not a bone structure in a cell, but it's going to help maintain the cell shape, create anchors and places for moving things. So let's look at the cytoskeleton in a cell can relate to two structures that you probably are familiar with already, cilia and flagella. So these are really extensions of the cytoskeleton. So if this is the plasma membrane here, you have these proteins that are extending out. In the case of the flagellum, it comes out. There's a singular flagellum in human cells. It's just the sperm cell, which you're seeing over here. And these proteins are able to switch back and forth, kind of rotate around and move the cell. So both cilia and flagella are used in movement processes. Both are made of tubules that are part of this cytoskeleton. The cilia are very short, and you're seeing here cilia that are lining, in this case would be, for instance, lining the fallopian tubes in the female system, are all these cells with cilia. And here's the the sperm cell that is swimming along that tube looking for an egg to fertilize, if it could look, if it actually had eyes. All right, moving on to the organelle known as the mitochondria. We're going to take just a few minutes to talk about energy and how energy is produced in the cell. Main thing is to understand mitochondria are the big energy producers. Producers of ATP, our high energy molecule, sometimes called the energy currency of the cell. So this is what they look like. Here's an actual mitochondria with all the foldings. So what are they important for? A process called cellular respiration. So we know that inside the mitochondria are enzymes that help cells to do respiration. You think respiration, you think breathing, you think oxygen. So in fact, oxygen is going to be used and CO2 is going to be produced. And that happens in the mitochondria. So of course, this is why we need to breathe, to use the oxygen in the production of ATP, and CO2 is going to be a waste product that we exhale. The important thing to understand about the mitochondria, there's probably more, not probably, there's a lot more detail here than you need to understand. Let me pause for a minute. Here is the slide where you are seeing, it's hard because it's not labeled here. This is a blood vessel here. All right, BV blood vessel. So blood vessels are bringing oxygen. Oxygen comes to the cell and enters the cell. Well, the cell has these structures called mitochondria. Here's one over here. So mitochondria, as we mentioned, are important for cellular respiration. What are we cranking out? Lots of ATP. So the only thing to recognize would be that inside the mitochondria, we're actually using the oxygen, releasing some CO2, but the process of getting the fuel in there, it involves multiple steps. One step is called glycolysis, where we take a, what is this, glucose molecule, and we break it up. And then that is moved into the mitochondria, where two other processes occur, one called the citric acid cycle. You might have studied the Krebs cycle. So glycolysis happens first, goes on to a process in the mitochondria called the citric acid cycle. And the third thing is this thing called an electron transport chain. Just simply recognize them as the steps involved in producing lots of ATP in the cell. If oxygen is not available, we can't complete this process, and that is a process that we call anaerobic respiration. So here's the, really, again, the summary. Cellular respiration using glucose and oxygen, producing water and CO2 as waste products. These are the three steps that are involved. And that's the bottom line. Lot to memorize. And finally, can we make ATP if there's not enough oxygen around? Well, we can do that for short periods of time. So an example is the process called fermentation. So fermentation is also can be referred to as anaerobic respiration. Anaerobic tells you an without, aerobic referring to the availability of oxygen. So when there is not enough oxygen available, we use the process called fermentation, or anaerobic respiration. Occurs not inside the mitochondria, occurs in the cytoplasm of the cell, does not require oxygen, involves the process called glycolysis. Remember, that was happening outside the mitochondria. You don't make much in the way of ATP. So that's the bottom line. You'll get a little bit of ATP. You don't need to have oxygen. This is only going to happen for a short amount of time. So sprinting, lifting heavy weights, things like that, where the muscles are contracted, using up the ATP quickly, so you can't maintain, if there's not enough oxygen, you can't maintain the production of ATP. For example, again, with sprinting, 100 meters sprint, that sort of thing.